In this video, we start with a completely new set of multivariate techniques. And these are distance-based, so that's different from what we had before. Before we covered all the rotation-based techniques. But in some instances, they actually try to accomplish the same thing. So we have ordinations again, and the other large set of methods that we're covering here are clustering techniques. And all of these techniques consist generally of two steps. So you first calculate distances, and there are many to choose from. So that's actually a conscious choice that you should make. You have to calculate your distances between points or observations or unit. Then you select the clustering method. So these always come with a default distance to use, but normally that would be a conscious choice. So you shouldn't just run a clustering method or an ordination method without thinking about your data and your distances first. So I'd like to start by looking again at our toolbox where we are. So we already covered many of the rotation-based techniques. We had the principal component analysis and the factor analysis here that we covered. We did multivariate analysis of variance, discriminant analysis, and we used it for different purposes, so you can make a prediction to which group a particularly new observation belongs, and we could analyze what actually differentiates classes. But what the rotation-based techniques couldn't deliver is actually a method to actually make those classes. So this is what we're going to cover today. So we'll do a cluster analysis here that actually develops a classification system. We also use those distance-based ordinations to reduce complexity. So we'll cover nonlinear multidimensional scaling here. We can also put MDS here. So that's metric dimensional scaling. So that also goes by another name, PCOA. So principal coordinate analysis. These are equivalent. And so the objectives that we cover with this first set of videos is complexity reduction and classification. So this is just another set of ordination techniques that we now know what that is. For the cluster analysis, we build dendrograms, so that's a new way of displaying multivariate data, so we'll cover that as well. But we'll go through all of those, so there are equivalents to rotation-based techniques for analyzing treatment effects, for making predictions, and for understanding relationships between two data sets, so we haven't covered any of that yet. So there are still a couple more videos to come. So let's do a brief recap of what we're trying to do. In ordination, we usually have a multivariate data set that looks like this. So my observations are in row. Those could be sample plots or individuals or locations. And then we have a whole bunch of measurements, multivariate measurements, that could be climate variables or soil variables or anatomical variables or species counts, whatever it may be. And we want to simplify those in a multidimensional plot. So if we have only three variables, we can actually visualize this as 3 dimensions, but they are projected here on a 2-dimensional space. And let's say I only have 3 variables in 3 dimensions and 3 points. Then this is a solvable problem. I have no issue with this kind of thing. So you could imagine if I do a PCA, that's a rotation-based technique, I can simply twist this coordinate system around to get a perfect representation of that triangle. So I have distance from here to there, from here to there, from here to there, and these distances can be perfectly represented on a 2-dimensional plane. So if you were to actually run this little table here with the principal component analysis, you would see that 100% of the variance is explained in 2 dimensions. So that's no problem. I can then actually add my vectors, and that 3-dimensional data set would be perfectly described in 2 dimensions. But typically I have lots more than just 3 observations, and then things becomes a little bit difficult. So for example, if we think of a tetrahedron, so 4 points here, I just added a fourth point that I want to display in this 3-dimensional diagram. So no matter how I'm going to do this, there is no solution for this, right? So these distances are always going to be too short. This one is too long. If I twist it in another way, that doesn't help. So this one is definitely too short. there's nothing I can do to get this right. In distance-based multivariate analysis, this is called stress. So I'm not able to honor all the distances. So I've done a job here that's as good as I can get it. There's simply no way that I can get this perfectly right. So if I look at a complex data set like the iris data set that we used before, then clearly there is a lot of stress in those ordinations. So if you think of this as a rotation to be a principal component analysis here, with my vectors being the original axes. I said before that the way you interpret these vectors is that all the points that have high sepal length are perpendicular to this sepal length vector. So they would all sit here along this in a perpendicular way. So the biggest sepal length are here, the smallest sepal length are at the other end. And the same for petal width, and the same for sepal width. But there can certainly be a lot of stress in these graphs. They are just imperfect representations. So if I were to draw drop lines for 2 points to see where they really sit, it could, for example, be that this point here is way in the back, so that sits right here. And this point here could be way in the front, so I can't really see that particularly well from this angle. So even though those points look like they are very close to each other, and the distance, if I were to put it in here, that distance looks as if it's very small, but with those drop lines you can see that distance is actually quite large. So those rotation-based techniques often have quite a bit of stress in them. Now I can do slightly better. So what I can do is I can think of my data as being on a rubber sheet, and I can unfurl this sheet in any way I like. So if I drop all my points on a curved sheet here, if I have it in 2 dimensions this way, then I could potentially honor many more of those distances correctly. So this sort of non-linear, non-rotation-based transformation could yield better representations of my relative positions of those points with less stress. So that is something I could not accomplish by just looking at this data set from different angles. So we lose that orthogonality, so my axes are not at right angles anymore necessarily, but I get better representation of the distances. Or I can think of completely alternative distance visualization. So that's a cluster analysis with a dendrogram. So my points or observations become leaves of this inverted tree, and then the ones that are close to each other, there you can read off what their distances are, literally those. So those distances here are represented by this distance scale, and as my observations become far apart, for example the blue and the reds, so then those distances are higher. So how do we calculate these distances? So any of these techniques require that we convert our data table into a distance matrix. And the way this works, so this is my normal data table, and I do them one by one. So I'm looking at my plot ID 1A and my plot ID 1B, and that may be a green point and a red point. And I have my multiple variables, and I look at the distances here, so 1 and 2, 1 and 3, 2 and 3, and I can calculate that distance and enter it here between 1A and 1B. So that would be my first spot where I want to enter that distance. And the distance calculation is quite simple. So that's a Euclidean distance here. So if I have my point 1A, so that would be a 1 and 1 on variable 1 and 2. And my point B is 2 and 3, so that sits here. Then I have a right triangle. I can calculate the hypotenuse, so c square, with the Pythagorean theorem. And so I just take the square root of this, and if I plug this in, so my distance here is 1 squared plus 2 squared is 5 squared is 2.24. And so this was just for the first 2 variables. But that one is actually very easily extendable, so Pythagorean theorem works also for a 3rd dimension if I were to put this in here. So all I need to do is add my additional distances. So in this case it would be another 1, so the difference between 2 to 3. So that would be this number here, and I come up with 2.45. So I can put that first number in my data table, 2.45, right here. and so on. So I work my way through the table and calculate all the distances. And of course there are functions that do that for you. So the general formula for Euclidean distance is that you take one value, so let's say my variable 1 is x, so it's a coordinate of my first point minus the coordinate of my second point. I square that, and then I take y, so that will be my second variable. Same idea, so coordinate of the green point minus the coordinate of the red point, squared, and so on and so forth, and then I take the square root of all of this. So that's a widely used distance metric. It's almost always a default in distance-based ordination and clustering, but there are others. So another one that's important is the Mahalanobis distance, and that's the same thing, a Euclidean distance, except that you do it on principal component scores. So you first run a principal component analysis, your PC1 and PC2 become now your x and y and z and so on variables, and you calculate the distances based on this. So that has advantages. So if you have autocorrelated variable sets, what you're really doing here, you're calculating the distance among independent dimensions. So for example, if I have 5 or 6 temperature variables that are really very highly correlated, I don't really want them to dominate my ordination just because there are many of those. So if they are 100% correlated, as an extreme example, they would all count as one variable in the end. And only the independent dimensions, so if they are not correlated at all, then they count as separate variables. So if 2 variables that are 0% correlated, PCA will not summarize them and they count as separate variables. But as you recall from the PCA, so there's lots of gray areas in between. So most variables are somewhat correlated to others. So PCA does those summaries for me, and then I can do the Euclidean distances on my main independent dimensions. So that's a very good distance to use. Almost all our packages will have that one as an option. And they're also standardized. If you recall the principal component analysis, if you set the correlation matrix to true, then all variables are normalized, so, and then all variables are also weighted equally. So that's not necessarily the case if you use Euclidean distance. But that's also something you could do before the Euclidean distance calculation. You could standardize your variables. So if you have different types of variables, that may be something you want to do. And then the third important distance measure is the Bray-Curtis distance. So that's good for binary data and any kind of zero-inflated counter or abundance data. So that distance is widely used in community ecology, where you have presence-absence data of species, or frequency or abundance data. And that's also quite simple and quite clever. There are 2 versions of this. So one is the Sorensen distance, so that works with 0 and 1s. So if I were to calculate that distance just for 2 points, so you can think of this plot A here that I use as an example as 1 point and my plot B as the other. So if I wanted to calculate the distance based on multiple species, so my variables are species, presence or absence. So it's the number of species that both plots have in common times 2 divided by the number of species in plot A and the number of species in plot B. And the Bray-Curtis version is exactly the same kind of thing, except it does work for count or continuous data, so it doesn't have to be 1s and 0s. So in that case, it's the sum of the lesser frequency of species in A and B. So if one plot has a 0, then my value for that particular variable becomes a 0. And let's say I have 90% in plot A and 80% in plot B. So what you would sum up here would then be the 80% for that second variable, and so on. And then you divide by the sum of frequency in plot A and the sum of frequency in plot B. So a nice property of this Bray-Curtis distance is that the values always range from 0 to 1. And if you think about it, that makes sense. So let's say that I have 10 species, 10 variables, and plot A and B have 6 of those in common, and the other 4, they have both zeros, so they're exactly the same species composition. What I would get then here is 6 times 2 is 12, and if I divide that by the total number of species in A, that's 6, and total number of species in B, that's also 6, so that's also 12, 12 by 12 is 1. So that gives me the 1 equals identical metric. And if they were completely dissimilar, so let's say 4 species are in plot A and the other 6 species are in plot B, then the number of species that are both in plot A and B, those will be 0. So 0 times 2 is still 0. And then if I divide it by whatever number, it stays a 0. So 0 would be maximum dissimilarity. And it works exactly the same way for the count or continuous type. So that's a great metric that works for pretty much anything. So if you have highly non-normally distributed data, zero inflated data, big time outliers, this is generally a very well behaved distance. And in R, so that would be the first manual step that I would recommend, is just to work with the data set that you have, so with the variables that you actually want to use for calculating your distance. So let's say they're in column 3 to 5, and then you just calculate those distance matrices, and you can specify what they are. So there are a whole bunch of other ones. I find these are the most useful. But if you're working in particular fields of research, you will encounter custom distances that are used in your particular area. So for example, in genetics, you don't use these kind of distances because you're working with genes that are usually diploid and they have alleles. So if I want a genetic similarity between two organisms, I need other distance measures. So for example, there's Ney's genetic distance, or there's a Jacquard distance. So usually in your particular field, you will have distances that you can import and then work with those. You can use all the clustering techniques or ordination techniques with any kind of distance matrix. So you should always think about this as a two-step process, and this first step should be under your control. So the last thing that people do is sometimes they square the distance matrix or take the square root of the distance matrix. So if you have a distance matrix with lots of very small distances and a few large distances, then you can take so you kind of have a skewed distribution of differences. Then you could use a square root transformation in a way to have a more even distribution of distances. So that makes your dendrograms or plots a little bit more pleasing. Or you can do it the other way around. So if you're in the other situation that you have lots of large distances and few small distances. And last but not least, I want to mention a couple of troubleshooting tips. So sometimes when you calculate those distance matrices, you will get errors for some reason, depending on the distance metric that you're trying to calculate. Or once you have the distance matrix and then you try to apply a technique, you get some weird error that you just can't figure out. And the way to troubleshoot this are the following things to check. So first of all, you should look at columns that are all identical. So if I have a large species matrix and maybe I work with a subset of my sample points, there may for some reason be a column that has only zeros in it. That is usually something that either the distance matrix or the clustering technique doesn't like. So throw those columns out. That would be your first check. And you can do that without any problem. So if you just have a column of zeros or a column of ones, so as long as they have identical values within a variable column, for all the plots or for all your observations, for all your individuals, there is really no information in there, right? So toss those columns out. That's the first step. The next one is remove all rows that are zero. So that can actually mean something. I don't like to throw out rows that are all zeros because I may have plots that just have zero frequency of a set of plants in it that I'm interested in ordination. So you can toss them all out, or another thing that you can do is set all your zero values to very small values. So if you have a species count 1, 2, 5, 10, it doesn't really matter for your ordination whether you put a 0 there or 0.00001. So in practice they will ordinate exactly the same. But for computational reasons, either your distance metric that you choose or the ordination technique cannot handle zeros. So you get some sort of division by zero error. So that can be fixed by a simple search and replace in Excel of all zeros to a very small value. And then the last trick that you can use. If you have identical rows and columns, that's also something that some techniques or some distances they just stumble on. They have some sort of division by zero, something does not work out. So in that case, if you have identical rows and columns, you can edit an observation to add a really, really small value, and then everything will work all of a sudden. So those in the order of importance and reluctance to apply them, those would be my troubleshooting tips. So that for sure you should do that in anyways, because there is no info. This would be the second level of manipulation to get your techniques to work, and that would be the last resort if you're still unsuccessful. But usually if you go through it in that order, things are most likely resolved just after the first step. So you could try that out with the first section of Lab 5 if you want. Or you can continue watching the videos on ordination and clustering before. So this is all covered in Lab 5.